learners. Here I have taken some previous year question from the exam UPPGT 2021. Subject is biology. A very important conceptual question I have selected here. Let's read it first. In Griffith experiment of transformation, virulent strain is called as, options given are S strain, R strain, M strain and K strain. क्वेश्चन कह रहा है कि इन ग्रिफिथ एक्सपेरिमेंट ऑफ ट्रांसफॉर्मेशन, ग्रिफिथ का जो एक्सपेरिमेंट ऑफ ट्रांसफॉर्मेशन है उसमें वायरुलेंट स्ट्रेन को क्या कहते हैं. ऑप्शन जो दिए हैं वो क्या क्या हैं. एस स्ट्रेन कहते हैं, आर स्ट्रेन कहते हैं, एम स्ट्रेन कहते हैं या फिर के स्ट्रेन कहते हैं. तो इस क्वेश्चन का आंसर करने के लिए हमें क्या जानना पड़ेगा. सबसे पहले हमें ग्रिफिथ एक्सपेरिमेंट के बारे में समझना होगा कि यह है क्या. लेट्स चेक द ग्रिफिथ experiment of transformation. Now Griffith experiment, this experiment is also known as principle of transformation. Alright, principle of transformation. You will understand later why this experiment has given this name. This experiment was conducted in 1928 by Frederick Griffith. He was a medical officer and he had developing vaccine against the disease pneumonia. For this he has conducted several experiments in a row. You know, pneumonia is caused by bacterium Streptococcus pneumoniae. Now coming back to the experiment conducted by Griffith, what were those experiments? Let's see another slide. Here he had cultured the bacteria Streptococcus pneumoniae, which causes pneumonia in the mammals. He cultured these bacteria on a culture medium and observed two types of colonies. One colony was named as S colony and another colony was named as R colony. The name given to these colonies is because of their texture. S colony is smooth in texture and R colony is rough in texture. The reason behind smoothness of S colony is that it consists of outer capsule which is mucilaginous, that is why it is smooth and shiny, whereas R colony is lacking this outer capsule. It lacks the outer capsule, that is why it is rough in nature. Now coming back to the experiment conducted by him using this bacteria, which is Streptococcus pneumoniae, having two types of colonies. In one set of experiment, I have named it as A. One A set of experiment, what he did, he took S strain of the bacteria and injected into the mice body. Results were, you can see mice dies. Now understand this, strain is nothing but variety of bacteria. R strain, R variety of the same Streptococcus pneumoniae bacteria were injected into the mice body and result seen was the mice were live. Now the reason behind, we have to understand the reason behind these observations, why mice was live and why mice died. For this we have to go to another slide here. Now S strain, what is happening here? S strain causes disease, that is why the mice dies, right? In the previous experiment set In the previous experiment, set A, these strains are therefore named as virulent or pathogenic strain of bacteria. And R strain, we have seen that when they are injected into mice body, they do not cause any kind of disease. Mice survives here. Therefore these strains, the R strain, are avirulent, non-virulent or non-pathogenic. Any term can be used here. Alright, now we have seen that S strain, mice dies, and die, mice is plural and mouse is singular, R strain, whereas R strain injected when into mice, the mice live. Here now we have understood why the mice die and why mice live, because S strain is virulent in nature. When it is injected, it will kill the mice. Now what is the reason behind the virulence of S strain? Why it is virulent? Why it is pathogenic in nature? Why it causes pathogenic means why it causes disease? Because since it can cause disease, the reason is because it can resist the animal's immune response, therefore is able to cause disease. And the reason behind its virulence is that its outer capsule helps it in protecting itself from the immune response. Alright, whereas R strain is non-virulent. We have seen it is non-virulent, non-pathogenic in nature, non-pathogenic in nature. What's the reason behind its non-pathogenicity? Since it lacks the outer capsule, and we know that outer capsule protect the bacteria from the immune response of the mouse body. Alright, since it lacks it, therefore the mice body, when it is injected, these bacteria when injected into mice body, the mice immune response kills the R strain of bacteria. Therefore they are not able to cause any kind of disease to the mouse. That's why they are non-pathogenic. Coming back to another set of experiment which I have named here as B set of experiment. Alright, in this set of experiment, Frederick Griffith used the technique of heating the bacteria to the extreme temperature. Extreme temperature makes the S strain of bacteria ineffective, and when these heat killed S strain of bacteria injected into the mice body, surprisingly the results were quite opposite from the A set of experiment. Here mice live. Again, when heat killed S strain, the same heat killed S strain is mixed with live R strain, and this mixture is injected into mice body, then the results were that these mice die. Alright, what is happening behind this? Let's check it out. Here we have seen that heat kill S strain is not effective, it is ineffective in nature. means it cannot cause any kind of disease. And when it is mixed with R strain, we have seen that R strain, live R strain, can't cause any kind of disease in the previous experiment. Keep this thing in your mind, that R strain can't cause any kind of disease because of lack of the outer capsule which protect it from the immune response. Now what is happening here? Heat killed S strain and live R strain, when mixed together, we can see in the previous slide that when these are mixed together, they cause the mice to die. Alright, it means something must have happened with the R strain which was initially avirulent, or it was not able to cause any kind of disease, but when mixed with heat killed S strain, it causes the mice to die, it causes mice to have that disease. Alright, now let's conclude few more things here. Griffith in the same experiment explained that his conclusion was like when heat killed S strain is mixed with live R strain of bacteria, R strain somehow got transformed by the heat killed S strain. This was his conclusion, and he added here that S strain becomes virulent as it starts synthesizing the outer capsule. Which is made up of polysaccharide coat. Here what he said, due to transfer of some genetic material it is happening, but he could not explain the biochemical nature of the genetic material which was getting transferred from heat killed S strain to live R strain of bacteria, which was causing the R strain to become virulent. Now coming back to the question asked in UPPCT exam, in Griffith experiment of transformation. Now let's solve it. Virulent strain is called as, now virulent strain is that strain which can cause pathogenicity or disease, which can cause disease in the animal. You know very well now that virulent strain is nothing but the S strain of bacteria. R strain is avirulent, S strain is virulent or pathogenic. We have came across with these two strains. We have not seen the name M and K strain Griffith experiment. So the answer you have to choose from these two options only, for which you have to understand the concept so that you can remember the process to answer this question. So this was the explanation of this question. I hope this helps you.